presentation is called Do More with Less. Because the technology that we will be seeing today will allow you to reduce time, to reduce sample volume that you are using for your studies, but also while keeping the high throughput information actually for your studies. So first of all, we'll start with a little bit of a background about biomarkers. So since this is a technology, or the multiplexing technology is a technology used to study biomarkers, so proteins, biomarkers, so based on a definition built by the National Institutes of Health in 1998, is a feature that is objectively measured and evaluated as an indicator of normal biological processes, pathogenic processes, or pharmacological responses to a therapeutic intervention. What does this mean? So biomarker is actually a medical sign that will always give us the same information about a specific physiological state, give us every time an accurate information, and will lead to almost the same conclusions every time. So how many types of biomarkers are there in the field? So there are genomics, transcriptomics, like the ones that Shenzi was talking about. There are proteomics as well, and metabolomics biomarkers. The mostly studied one remains proteomic biomarkers, because they are quite accessible, there are plenty of techniques that can be used actually to study biomarkers. In our case today, so disease-related biomarkers, what can they provide for us? What is their role? So they can help researchers or clinicians as well to detect the disease. They can help identify the disease stage, like in our case today, cancers. It can help predict the response to a treatment, so resistant, sensitive to the treatment, determines the efficiency of a treatment. It checks the compliance of a treatment, because sometimes some treatments might lead actually to adverse effects. And of course, there are some biomarkers that can be used to identify that adverse effect. And of course, it helps control the progression and the recurrence of a disease. And of course, and again, in this case, so today for us, sorry, I'm just lowering a little bit the camera. For us today, so of course, it's controlling the progression, and also the relapse of cancers is very important. So biomarkers, of course, will help improve the treatment, because whenever identified, and identifying the differences and this transition from the healthy state into the advanced disease, or even the early stages of a disease, will help clinicians and researchers improve the treatment as well, and improve sometimes and lots of times now the life of the patients. But actually, identifying this transition might not be always that easy, because lots of sensitivity is needed to identify actually the concentration of different biomarkers, and which is not always easy with the available technologies. But as you know, lots of techniques and lots of technologies are available to study biomarkers. Oh, sorry. We can start from Western blot, which is also was mentioned by Shenzi. So very basic technique. So it's a qualitative technique, into the more advanced ones like LC-MS, MALDI-TOF, which are very expensive technologies and needing lots of expertise, but also a lot of money. And in between, there are plenty of technologies that can be used for biomarker studies, but we can start from ELISA, for example, which is a very well-known technique. We can go to multiplexing until up to the high sensitive or ultra sensitive technologies that we as Merck offer, like the single molecule counting technology. So we'll move now to what we offer as a company. So like I mentioned, so we have technologies in between. so cost-effective, but also that allow customers and researchers to have more reliable, more results than more parameters, and to make more conclusions than the techniques like Western blot, which in this case, so ELISA, which is a technique, like I said in the previous slide, we've seen in the previous slide, is so it's a basic technique. It's very well known, but ELISA has several limitations that we will see later on, which can actually reduce the field where researchers can play. There is also the multiplexing technology that is coming from a company called Luminex, and this is a technology that allow actually the detection of up to 500 biomarkers or proteins at a time. So and also the single molecule counting technology, which is a highly sensitive technology that allows the detection of proteins with a concentration that goes down to the femtogram, 10 times 8 minus. So this is a very sensitive technique, but it's dedicated to specific studies, like, for example, drug development. So, okay, we've seen ELISA. We said it's ELISA. We know that, and I personally know that lots of researchers are still comfortable with the ELISA technique, but of course, I will always recommend researchers to use ELISA if they are looking or studying one biomarker. So one ELISA kit will be sufficient. If they will be looking for multiple biomarkers profiling, then of course, can you imagine your lab being drowned under all these ELISA kits, which is not a convenient solution. So for that, so Luminex, the mother company of the multiplexing technology, came up with a solution called the Xmap Technology. And in this slide, we will see the difference between the two technologies. So the Xmap and ELISA. So let's say that Shenzi, for example, is working on 10 proteins, 38 samples. So if Shenzi will be using ELISA as a technique for his study, so he will need 10 proteins, so he will need 10 different kits. The total time to result for the analysis would be at least 10 hours. The results calculate will be 38 because he will be having results for each protein for 38 samples or patients. The volume needed per sample will be around 100 microliters. So the total sample volume used for the study will be 1,000 microliters per each individual or sample. If Sanchi will be using ELISA, then he will not have internal controls for the assay, and the dynamic range of the technique goes between 10 picograms and 2,500 picograms per milliliter, and the lower limit of detection is around 10 picograms per milliliter. If Shenzi then decides to use the Xmap technology, then of course everything will be put into one plate. This will bring down the time needed for the results to 3 hours. The results per plate that he will be getting will be 380 because he will have information for the 38 samples and the 10 proteins from one single plate. The volume needed per sample for this study per individual will be 25 per samples, will be 25 microliters, and the total sample used for the study per individual or per patient will be 25 microliters because that's the volume needed to run a sample on to run on the multiplexing assay or platform. Internal controls possible, yes. So he will know if things doesn't go well. He will know where actually if it's him not being concentrated while manipulating, or it's us. It's our problem. The dynamic range of the technology is 10 times higher for the lower limit of detection and 4 times higher than ELISA for the upper limit detection, lower limit of detection and the upper limit of detection. Yes, and the lower limit of detection of the Xmap technology is 1 picogram per milliliter. I really like this slide so much because it summarizes the difference between the two technologies. And here you can already start visualizing everything that you will be saving, actually, time, samples, money as well. So budget, I mean, for the study. So, okay, let's go back now to the basics. So where does this technology come from, and how is it built, and how does it work? So the technology comes from, like we said, so Luminex. So the mother company of the technology, it used to be a United States and American company, but now it has been brought, like lots of companies, so by another bigger company, so DiaSorin, and it's now a European company. So it manufactures the magnetic beads that we will see in the next slide are the basics, or the base of this technology. It manufactures equipment, and we as Merck, so partner for technical support, and also we sell the machines, for technical support of the machine. And we partner to build our own assays, which are the Milliplex kits that we will see later on. So DiaSorin, the new company for the broad Luminex. So what does the Xmap technology mean? So X for unknown, M for multi, A for analyte, and P for profiling. So it's a technology that allows the multiple analytes, unknown analytes profiling. Of course, 4 basic components are needed for the Xmap technology, which are biological reagents, the magnetic microspheres, the detection instruments, and of course the software for the acquisition of the samples and the analysis. So Luminex was founded actually in 1995, and since then they developed bite by bite the portfolio. So they have started in the very beginning with the Luminex 100, then moved into the Luminex 200, and then to the FlexMap 3D, and then to the MacPix in 2010, which they have discontinued because, of course, as you know, everyone is struggling because of COVID. So yeah, so they had to remove that instrument. And very recently, so last year, they launched the Xmap Intelliflex and the Xmap Intelliflex DRC. So which is actually the next generation instrument for the multiplexing technology. So the Xmap technology's secret is those little beads that you see here. So they are 6.5 micron microspheres. So they are, if you can picture a red blood cell, so they are smaller than a red blood cell. So they are so tiny, and they are made of polystyrene. So, and the polystyrene actually is a chemistry that allows, because it has carboxyl, how do we call them? Carboxyl groupments. So they allow the binding of oligos, of antibodies, of antigens into the surface of these beads. So very tiny, small, very tiny beads, magnetic beads, so which are internally color-coded. So they have code inside of them in order for the machines that they will be running on to recognize these beads. So they, for the Luminex 200, these beads are color-coded with colored with 2 dyes, 2 different dyes at 2 different proportions. And for the FlexMap 3D and the Intelliflex, the more advanced equipments, so they have 3 color codes and 3 dyes inside of them that will allow to increase the number of combinations. So the basic, the LX200 instrument, because of that color code, so it allows the detection of 100 biomarkers or proteins or DNA sequences, RNA sequences at a time. The FlexMap 3D and the Intelliflex, which are the more advanced instruments, can detect up to 500 biomarkers, proteins, or even like I said, DNA or RNA fragments. So these beads are color coded from the inside, and of course they are put together, of course, in a solution, and they are later on being analyzed. And briefly, in this case, for example, we have if we say that for IL-6, so for beads that will detect the IL-6 protein, so they have a color code with, let's say, 90% red dye and 10% infrared dye. For IL-1, let's say 80% red dye and 20% infrared dye, and so on and so forth. So this allows to multiplex, so each bead will represent an ELISA plate. This will increase actually the surface, the contact surface, so that is used actually to detect biomarkers, and of course allow the mixing of millions and millions of beads at the same time in the same well. So how does it go? So the assay overview compared to ELISA. So the principle is the same of the ELISA, but like I said, so the beads will play the role of the plate. So first of all, we have the stock microspheres that have the carboxyl binding sites on the surface, as you can see here. So the capture reagents, so either antibodies or oligos, are coupled to the binding sites on the microspheres. Then the sample is added to the microspheres and bound by the capture reagent. And of course, the last step will be adding the detection antibody into the fluorescent reporter, so which binds to the analyte of interest, and then everything will get. This complex will get excited by lasers, and then the lasers. So the beads, so they get excited by a green laser and a red laser, and of course the fluorescence emitted by these beads is going to be captured by the instrument and analyzed. So the Luminex instruments that are now still alive in the market, so the Luminex 200, the Xmap 3D, and the Intelliflex are flow cytometry based instruments. So they have lasers inside of them, and so like I said, so the beads with the protein of interest bound into its surface will migrate in front of the lasers one by one and will be analyzed one by one. Of course, the red laser will interrogate the bead, and in order to classify the bead, to know what is the proportion of the dyes inside of the bead, and of course the green laser will then identify or quantify what has been bound into the surface of the bead. And that's how we have then the information that we are looking for. So, of course, like I said, so these beads will have to be run into inside of an equipment, of course, because even for ELISA, we have to use the ELISA reader. So these are more sophisticated ELISA readers, if I may say so. So Intelliflex is the newborn of the multiplexing technology and of Luminex. So if you compare a little bit, and for the ones who already have an idea about the multiplexing technology, so the other instruments like the LX200 and the Xmap 3D, so they are, you'll see the analyzer, the computer, you have underneath, so you will see the sheet fluid, which is the liquid that is, and the fluid that is used to transfer the samples inside of the equipment. But here, so Luminex moved into a more compact instrument, a more compact and modern design. So here you see, like, it reminds me a little bit of the real-time PCR analyzer. So you have the instrument, you have the computer, which is embedded into the instrument, and of course, everything needed so to run to get the results, everything is in the equipment. And like I said, so this is an equipment that allows the detection of 500 biomarkers at a time in for a 96 well plate, so it takes 20 minutes to get all the information, all the results. For the 384 plates, it takes a little bit longer, so it's around 75. If the information is not correct, we will see it in the next slides. So when we will compare the different instruments. So yeah, it's quite an. I keep seeing. Okay. Keep seeing the names of the participants to the right side, but yeah, oops, sorry, I managed now to remove the bar. So yeah, okay. So like I said, so it's a compact instrument. It will not take a lot of space on the benchtop, and it runs 96 well plates, 384 well plates. And of course, I mean, everything needed to run the instrument itself is in the instrument, except the sheet fluid, like I said, the fluids that are used to transfer the samples inside of the equipment. So will have to be put underneath the equipment, underneath underneath the benchtop. And of course, this instrument offers the widest dynamic range in the market. So it's more than 5.5 logs for detection. So yeah, here you can see so how it goes. So now this is your bead, and it goes inside of the instrument. So I'll go back again. It goes in front of the laser box. It gets excited by the red laser, and this actually the signal will be analyzed by 4 different photodiodes. So it helps the classification of the bead, so to know what proportion of dyes is inside of the bead, and the green laser then will excite the bead in order to quantify what has been bound into the surface of the bead. So the Intelliflex actually exists under 2 formats. So the normal Intelliflex research use only, and the GRSE. So the GRSE is the dual reporter side eject. So dual reporter, so means that on the surface of the bead, so you will bind your proteins, you will bind your antibodies, but there is a possibility to add another information that can be detected on the surface of the bead by using another detection antibody, a second detection antibody. So on the surface of the bead, let's say we have proteins, for example, so phosphorylated and non-phosphorylated proteins. So they will bind into the beads normally. So you will not be able to detect if these proteins are phosphorylated or not, and if you are using the normal technology. But using the GRSE, so there is a possibility to add into the existing detection antibody a second detection antibody that will allow to differentiate between the two type of proteins, which is actually something that increases the number of parameters or information that you can get from the instrument from 500 to 1,000, because then 500 different proteins, for example, and then for each protein you will have information about phosphorylation, non-phosphorylation of these proteins. Yeah, and this is explained in here. So, and this explains a little bit the schema. So how these beads are being analyzed. So you have the red laser for classification, and then the green laser for the detection of the first and the regular normal detection antibodies, and then the violet laser that will detect the second type of antibodies that are put in the solution. So yeah, different types of applications. So serology assays, for example, if you are looking to differentiate between, for example, IgGs and IgAs, free versus bound drugs. So the normal reporter, so you will allow you to detect, for example, if you are looking at immunotherapy, so monoclonal antibodies or whatever. So you have initially you can detect the presence or the absence, but now with the new version of the instrument and the technology, so you can identify if this protein or if this drug is bound into something else. So there is a possibility to distinguish between the two. And of course, the protein phosphorylation. You will be able to differentiate between non-phosphorylated and phosphorylated proteins. So, in a nutshell, for now, different instruments, different capabilities, of course, different prices and different output. So the Xmap here, as you can see, so the FlexMap 3D, Intelliflex, the Intelliflex DRSE, it's not coming soon, it's here already. So they have more capability. So if you have more samples to run, because they are run 384 well plates, if you are looking for more information and more parameters, they can go up to 500. And for the Luminex 200 is for really the lower throughput. So of course it can detect up to 100 biomarkers and 80 on the Maplex. Of course, like I said earlier, where is it? No, the information is not here. So we'll sit with 75 minutes needed to get the results for 384 wall plates. Of course, so we partner with Luminex to build the kits or the assays, all the panels that are run on the instrument. Of course, what we do is that we buy the beads, these colored magnetic beads from Luminex, and we provide all the rest from our portfolio. So we provide antibodies, we provide the capture antibodies, the detection antibodies, the reagents are needed to build the kit, and of course this allowed us to build a very wide portfolio for biomarker studies for different therapeutic areas, different research groups, and of course for different sample types and species as well. So overall, we have more than 100 kits in the panels in the portfolio, more than 1,000 soluble analytes, different species that can be studied with our panels for different applications like cancer studies, immunology studies, toxicity, neuroscience, cardiovascular diseases, etc., etc. Of course, since when building the assays, of course, we try to do our best to get for researchers to get the best results possible, the more reliable results, while guaranteeing efficiency, efficacy, everything. So with these assays, so we are, our assays and our manufacturing sites are ISO 9001 certified. Our assays are very well, I would not say validated, but verified, because when we say validated, it makes us think of IVD kits, but our kits, our panels are research use only. So we have this quality mindset that allows us to have to allow researchers to get the best results possible for these assays. And one thing that you will not find in the market is that the fact that of course, for these panels, you will need standard curve. So of course, you like just ELISA in order to extrapolate and identify the concentrations of your biomarkers. For us, one unique thing, we have one unique thing, which is that we provide real matrices in the kit. So for serum and plasma, we provide real serum matrix, real plasma matrix, which allows actually researchers to get the best results possible from their kits, because these real matrices mimic the condition, the natural conditions and the conditions in which the proteins exist in reality. And actually, even the American Association of Pharmaceutical Scientists has in the paper published in 2016 mentioned clearly that for drug development clinical trials, so immunoassays need to have at least this very important component, which is the real serum, the real matrices that will allow to get researchers to get more precise and reliable results. So, in a nutshell, again, so the portfolio is quite wide. So like I said, so we have panels for immunology, for cardiovascular diseases, for bone, for studies, for aging. So, and these panels are built in a way to allow researchers, first of all, so to pick any biomarker from the panel. So panels are designed or are put in that way in order to prove to researchers or to show to researchers that this is what they can combine from that panel. So these biomarkers are put together, they will not cross-react, they will have the same dilution factors. So they will the dilution factor, so no need to there will not be a need to worry about different dilution factors or whatever. So you'll just mix everything, put it in with your sample, and then you're ready to go. And yeah, cell signaling, Aziz, I know how cells talk to each other is very important, and for that, so we do have specific panels to study cell signaling, and these assays are specific for cell culture. So they are not like, sorry, I forgot to mention for the other panels is that they have been used on all kind of crazy samples: serum, plasma, cell culture, CSF, tears, milk, all kind of crazy samples. And we do have documented records of all the best practices, tips and tricks for these samples. Yeah, so, and these the second part of the portfolio, which is the cell signaling map assays, are exclusively for cell culture, for cell culture supernatant or cell culture study samples. And there is also a possibility besides the ready-to-use panels. So we have the possibility of mixing, matching whenever it's possible. So which we call the custom assays. So there's a possibility to mix between different panels. If you see that you are interested into 15 biomarkers, you found that 14 are in one panel, one biomarker is one in another panel. So it's such a waste of time and money. So we could check with our research and development team if we could mix those together in order to make one panel with 15 biomarkers. But also we can help you work on building panels that do not exist within our portfolio. So if you are you are hesitant, and you are suspecting that a biomarker can be used in diagnostics later on. So and doesn't exist within the panel, that of course we are more than happy to support you build assays for that specific biomarker or biomarkers specifically for the study. The good thing is that we have actually we produce antibodies.
Just 2 minutes left to do that. 2 minutes left.
Okay. Thank you. So we have we manufacture antibodies. We manufacture all the reagents. So we have this very good flexibility for building the assays. And besides that, we have even softwares that allow actually the analysis of these biomarkers of these panels. And even allow, the software, Belisa, can even help you analyze your ELISA results, not only the multiplexing results. Here, I will just finish with the Milliplex immune checkpoint protease panels, because I know that this is, it's quite important. So checkpoints are regulators of the immune activation, and they play a crucial role in the maintaining the immune homeostasis, the preventing autoimmunity, and also are involved in cancer immune escape. And of course, so for this, we have developed specific panels for the human immuno-oncology checkpoints panel. So we have two panels, 17 and 31 biomarkers for human studies or human samples, and we even have for anyone who's developing therapies. So we have one for mouse, actually. So the mouse immuno-oncology checkpoint protein panel 1. Looking for more information, I will be more than happy to share the catalog. Well, we have the Analyte Quarterly, which is the brochure for all the Milliplex and the multiplexing offer. Here, there's a specific page for the cancer panels. And you can go also to the website to get more information. And of course, feel free to reach out to me at any time. So I'm reachable via WhatsApp, email, and everything. So yeah, thank you very much, Mohammed.